Hello students, this is chapter number 11, Biotechnology Principle and Processes. And in continuation, this is video number 2. Previous video, we had studied about general term of biotechnology and as well as what are the basic principles, what are the steps taken in biotechnology. Now, taking it further, let us first study what are the tools of the recombinant technology. There are three tools. One is restriction enzyme, the second is cloning vector, and the third is competent host. So in this video, I'll be talking about restriction enzyme, the very first tool of this recombinant technology. First thing, let us understand what are restriction enzymes and why are they so called? Children, restriction enzymes are also known as molecular scissors. Okay, reason because they can cut the DNA. In 1963, two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. Okay. There were two enzymes, which was found that these two enzymes does not allow the bacteriophage to grow in E. coli, and these two enzymes were isolated. One of the enzyme added methyl group to the DNA and restricted the growth. It altered the DNA of the bacteriophage, and the other enzyme cut the DNA. And the one which cut the DNA, it was known as restriction endonuclease. From then, today, 900 restriction enzymes have been isolated out of 230 strains of bacteria. The first restriction enzyme that was isolated was HIM2. I have given here so many names of so many restriction enzymes. Now, there is a term known as recognition sequence. Let us understand what is recognition sequence. What do you mean by that? See, children, restriction enzyme cuts DNA molecule at a particular site. It's not कि वो कहीं पे भी cut कर देगा. So it cuts the DNA molecule at a particular point by recognizing a specific sequence of 6 base pair. You can see these are the sequences which the BAM-H1 recognition enzyme, restriction enzyme will recognize. So this is the recognition sequence. For example, EcoR1 recognizes a sequence of GAATTC. So this is GAATTC is the recognition sequence which is recognized by EcoR1. It is a sequence of 6 base pair, and this specific base sequence is known as recognition sequence. And then this enzyme will cut this recognition sequence at a particular point. Like EcoR1 cuts it between G and A. You can see the arrows over here. Next is these enzymes which cut the DNA at specific sites into fragments. They belong to a class of enzyme known as nucleases. Now, these restriction enzymes may be of two types, exonuclease and endonuclease. Exonuclease are the restriction enzyme which removes the nucleotide from the end of the DNA. ये DNA के बिल्कुल end से काटता है. Whereas endonuclease, it cuts this at specific position within the DNA. DNA के बीच में से कट करता है. DNA के बीच में से. They bind to the specific recognition sequence, as you can see over here. This is EcoR1. It will bind to this recognition sequence GAATTC at GA, and from here also it will bind at GA, and then it will cut at specific point. Cut the 2 strands at specific point. Now let us understand how to name the restriction enzyme. What is the process of naming the restriction enzyme? Children, students, the very first letter of the name comes from the genus. Like for example, this EcoR1 is a restriction enzyme. E comes from Escherichia. The second word C or O, CO, it comes from species. That is coli. And the third, that is the R, that comes from the strain. R word, the third word comes from the strain. That is EcoR1 comes from Escherichia coli RY13. So R is derived from the strain RY13. You can see over here. This is the strain of E. coli. So from here, the R is being taken. Then after that, EcoR1. What is 1? It is the Roman number. Is the Roman number following the names indicate the order in which the enzymes are isolated from the strain of bacteria. Means from this strain RY13, this was the first restriction enzyme that was isolated. That's why it is named as EcoR1. Now let us understand what do we mean by palindromic nucleotide sequence. Now, the sequence of base pair that read the same on the two strands when the orientation of reading is kept same, that is known as palindromic sequences. For example, GAATTC. Now, this reads GAATTC from 5' to 3'. Just see it's reading again in the second strand from 5' to 3', GAATTC. So you can see that it reads the same when the orientation of reading remains the same. So this is what? Palindrome. Now let's understand how does this endonuclease work, or what is the mechanism of action of endonuclease. See, you can see this diagram over here. There is a foreign DNA and then there is a vector DNA. And this is the gene which we have to transfer. So every endonuclease, first thing, every endonuclease inspects the entire length of the DNA sequence for a palindromic recognition sequence. Like EcoR1 will inspect the whole length and from where it will get its sequence, that is GAATTC. So on finding the palindrome, the endonuclease will bind to the DNA. So you can see over here the arrow showing binding to the DNA. It cuts the opposite end of the DNA in the sugar phosphate backbone a little away from the center of palindrome site, but between the same bases on both the strands. So in this strand also it will come from cut from GA, and here also see it's cutting from G, between G and A. So it will cut it in the vector DNA also, and it will cut it in the foreign DNA also. Now when it is cut, obviously you can see that overhanging stretches are produced. This results in the formation of single-stranded overhanging stretches. You can see over here, this is the single-stranded overhanging stretches. These are known as the sticky ends. Now, why are they called as sticky ends, or what are sticky ends? Let us understand both the things. What are sticky ends? That I have just now told you, that these single-stranded, after cutting, what is produced? A single-stranded overhanging stretches at the end of each strand is being produced. This is known as sticky ends. Now, why do we call it as sticky ends? These are named as sticky ends because they form hydrogen bond. Now, these two will come and they will get joined over here with hydrogen bond with the complementary parts. The sticky end facilitates the action of enzyme DNA ligase by readily forming a hydrogen bond with the complementary strand. Children, in genetic engineering, the DNA from different sources are cut with the same restriction enzyme so that both the fragments have the same kind of sticky ends. So in this video, I have talked about the tools, the first tool of DNA technology, recombinant technology, that is restriction enzyme. In the next, I'll be talking about the next tool, that is the cloning vector. Thank you.